Thank you very much for the introduction. So I'm going to talk about Stalk and NA-based next generation influenza virus vaccines that we are working on at Mount Sinai. But let me start out with a very brief introduction about influenza. I think in the last 2 years, we have mostly forgotten about the existence of this virus. Influenza viruses are part of the Orthomyxoviridae family. We have influenza A, B, C, and D, but only A and B are clinically relevant. These viruses have a lipid envelope, an RNA genome on the inside of that envelope. And this genome is segmented. We have 8 genomic segments for influenza A and B. One of the segments encodes for influenza hemagglutinin, which is the major surface glycoprotein on the virus. And this comes for influenza A in a huge diversity. We have 18 different hemagglutinin subtypes. And another segment codes for the neuraminidase, which is the minor surface glycoprotein, which also comes for influenza A in high diversity with 11 different subtypes. And then I'm also mentioning here M2 and conserved internal proteins because they are important for other universal or broadly protective influenza virus approaches. But we're going to focus basically on the HA and the NA here. Quick overview of influenza. In humans, the virus causes a spectrum of mild to severe respiratory disease. We have epidemics that are caused by influenza A and B, so-called seasonal influenza, and we have pandemics, which are only caused by influenza A. If you look at epidemics or seasonal influenza, before the COVID-19 pandemic, we had about 3 to 5 million severe cases worldwide and 290,000 to 650,000 deaths. In the US, the virus killed about 12,000 to 61,000 people every year. Of course, in the last 1.5 years, we barely had any flu cases. And then we have pandemic influenza. The worst example is the H1N1 pandemic of 1918, which claimed approximately 40 million lives. And these viruses are typically introduced into the human population from the animal reservoir, from birds or mammals. This figure shows the dynamics of influenza virus circulation in humans over the last 100 years. We start out here in 1918 with H1N1. The virus caused a huge pandemic and then became a seasonal virus. But because a lot of people got infected and had immunity, the virus had to escape that immunity, and it started a process called antigenic drift, basically small mutations, mostly in the HA, that allows it to escape from antibody neutralization. And so this virus then drifted until 1957. And then something remarkable happened. We had another pandemic, another introduction of at least partial avian virus into humans. In 1957, H2N2, and that's what we call an antigenic shift. And this replaced the H1N1 virus here. H2N2 circulated for about 10 years, drifted as well, was then replaced by another pandemic virus, the H3N2 virus in 1968. And this virus actually drifts quite a lot and is still with us. It's a major problem. In 77, H1N1 came back and circulated till 2009 and was then replaced by pandemic H1N1. And so we ended up with having an H1N1 virus and H3N2 virus and 2 different antigenically different influenza B lineages circulating in humans, and they change all the time. And so this is, of course, very complex for vaccine manufacturers, and this is the reason why the vaccine has to be reformulated every year and revaccinated. Now, if you want to understand what we are doing at Mount Sinai in terms of hemagglutinin-based broadly protective or universal vaccine approaches, we have to have a closer look at the HA. I have a monomer here on the right side of the HA. HA is the major glycoprotein of the virus. And we have a global head domain, which is membrane distal, and that mediates binding to host receptors. And then we have a stalk domain, which is membrane proximal, and that mediates fusion of viral and endosomal membranes. And the immune response targets mostly the immunodominant head domain domain of HA, which is great because these antibodies are potent neutralizers, but it's also a problem because this is where most of the antigenic drift happens. The Stalk domain is immunosubdominant, so it's not really targeted that much. What do we know? But the nice part about the Stalk is that it's quite conserved, it doesn't drift. So the good thing is that there are antibodies that do target the Stalk, and because the Stalk is conserved, these antibodies can be broadly neutralizing and broadly protective. So what do we know about these antibodies to the Stalk domain? They're not induced or boosted upon regular seasonal vaccination, but they have been isolated from humans and mice, and they bind to conformational epitopes that are relatively easy to destroy. They're HI negative, so you don't measure these antibodies in the main assay that's used to measure the correlate of protection for flu vaccines. Because of the conservation of the Stalk domain, these antibodies are broadly neutralizing, but they also have other functions. For example, they can protect via FC, FCR effector functions. And we know by now that they're an independent correlate of protection from H1N1 infection in humans. And I said that the Stalk is conserved. To better understand this conservation, we have to look at the phylogenetic tree of hemagglutinins. For influenza A, we have group 1 and group 2 hemagglutinins. Then a little bit further out, influenza B. We have major human pathogens like H1 and H2 in group 1 and H3 in group 2. But we also have these viruses or subtypes that sometimes cause zoonotic infections like H5 in group 1 and like H7 in group 2. And so typically these Stalk reactive antibodies follow this phylogeny because that indicates the conservation of the Stalk. So you have antibodies that bind broadly to group 1 or group 2 or influenza B. There are exceptions where you have cross binding and cross neutralization, but these antibodies are rare. Just to be a little bit controversial, I also wanted to show the diversity of the current diversity of SARS coronavirus 2, which is actually shown here with the Wuhan 1 strain, the Beta variant and the Delta variant. And of course, if you compare that to influenza, the diversity is relatively small. So we're dealing with a huge antigenic diversity here for influenza. So the question that we asked now many years ago was, can we induce protective levels of these broadly neutralizing antibodies by vaccination? And to do that, we designed what we call a chimeric hemagglutinins. I give you an example here. This is what we call a chimeric 6-1 hemagglutinins. This is a combination of an H1 Stalk domain, so a seasonal hemagglutinins with an exotic avian head domain. And these chimeric hemagglutinins typically have either H1, H3, or influenza B Stalk domains combined with these exotic head domains. And these constructs have three advantages. First, if I vaccinate animals with this construct, and then I challenge them with, let's say, an H1N1 virus, and they get protection, I can assume that the protection is Stalk mediated. In addition to that, the presence of the head domain stabilizes the conformational epitope in the Stalk domain. If you would just chop off the head, these epitopes would fall apart. There are technical solutions to that, but it's not that easy to make headless hemagglutinins. And finally, this is a fully functional hemagglutinins. We can rescue viruses based on this, which means we can make inactivated and live attenuated vaccines. But of course, you still have this immunodominant head domain present. Now, what we did with this construct was sequential vaccination. Basically, we vaccinated with one construct that had an H1 Stalk, and then let's say an exotic H9 head domain. And then and what this does is you get some immunity, some memory responses against the Stalk domain, and of course a lot of immune responses against the head domain. And then you can revaccinate these animals with another chimeric HA that has the same Stalk domain, but a different head domain. Now, again, you get a good response against the head domain, but it's a primary response. It's not a recall response because the animals haven't seen that head domain before, but they've seen the Stalk domain. And so you shift the balance a little bit to the Stalk because this is now a recall response, which is preferred by the immune system. And then you can revaccinate these animals another time with yet another chimeric hemagglutinin, which again has the H1 stalk, yet a different head domain. Again, you get a primary response against the head domain, and now a very strong recall against the stalk domain. And then if you would challenge these animals with H1N1, for example, you get very robust protection in animal models. And I just summed this up here because we have so many of these animal studies that we have published. These chimeric HA constructs induce broadly reactive anti-stalk antibodies and protect mice and ferrets from challenge with heterologous and or heterosubtypic virus strains, so within a subtype, but also across subtypes. We see that the observed protection is mostly antibody mediated. We see very good protection against emerging viruses. But what we have also seen is that we need a trivalent vaccine. We need a group 1 stalk, a group 2 stalk, and an influenza B stalk. And with a group 1 stalk, you basically induce antibodies that cover group 1, but they don't cross-react well. So you need all these 3 components. And then we went into a human clinical trial in 2017. And the idea here is a little bit different because humans already have pre-existing immunity to seasonal influenza viruses. So there's pre-existing immunity to group 1, group 2, and influenza B. And so the idea was that we only need 2 shots here in order to induce robust stalk immunity. And so we went in our first in human trials with a chimeric H1 construct, so an H8 head on top of an H1 stalk, followed by a chimeric H5-1 construct, an H5 head on top of an H1 stalk. And again, we think this is just a component. We think that we need a trivalent vaccine in the end. And so here's the trial design. This was a small phase 1 trial that was done together with PATH in Cincinnati and at Duke. A randomized placebo-controlled observer blind phase 1 trial in healthy adults with 65 subjects. And we started in December 2017. We had 5 groups. Group 1 and 2 received a chimeric HA-based live attenuated vaccine and then were boosted with an inactivated chimeric HA-based vaccine. One group got the inactivated vaccine with adjuvant and the other group got it without adjuvant. Now, why did we do this? Because there were several clinical trials that showed that priming with live attenuated vaccines followed by boosting with inactivated vaccines works better than just giving, for example, the inactivated vaccine twice. So we wanted to test this. We had a control group here, which received saline intranasally for the first dose and then BVS intramuscularly. And then we had another group that received the inactivated chimeric 8-1 vaccine with AZO3, followed by the inactivated chimeric 5-1 vaccine with AZO3. So intramuscular followed by intramuscular. And we had a control group here too. For the analysis that I'll show you, we lumped the 2 control groups together because there was really no difference. And this is basically the main takeaway from this trial. We were able, with a rationally designed vaccine approach, to induce these stalk reactive antibodies. What you see here in the purple line, this is the placebo control group. On the left side, we have serum IgG. On the right side, we have serum IgA. In green, we have the group that received the inactivated vaccine 8-1 and 5-1 twice with adjuvant. You see the first shot already induces a very high titer of stalk reactive antibodies. Then there's some waning. The second shot brings that back up. This falls, and from day 252 out to day 588, this is really stable. So we think that we can induce stable antibody titers. The live attenuated vaccine really didn't do anything. We think it didn't take, but then when the inactivated vaccine with or without adjuvant was given, we also induced the stalk response, of course, better with adjuvant. And this is mirrored in the IgA stalk titers in serum. There's many more results. I just don't have the time to go through all of this, unfortunately. But what I wanted to show you is breadth, the antibody breadth that we were trying to measure. Because of course, if you're trying to develop a universal influenza virus vaccine, you want to have broadly reactive antibodies. And so initially, and this is published, we looked at H9, H2, and H18 antibodies, but I really wanted to get a good feeling about breadth against all subtypes. And so that's what we did. We wanted to look at all subtypes. And we did that with a new analysis tool that we have in the lab that we call influenza virus protein microarray, or IVPM. And for this, we express different recombinant hemagglutinins. Then we print them on microarrays, which are then incubated with serum and then with secondary antibody. And then we read that out on a microarray reader. We do an analysis, and then we look at responses to these different hemagglutinins. The advantage here is you can look at a lot of different hemagglutinins at the same time with a very small serum amount. So it's relatively high throughput. And so this is what I'm showing you here. Again, we have our groups, the two live attenuated groups up here, the inactivated group here in the third row, and then the placebo group. And we have split this up here into group 1 HAs, group 2 HAs, and B. And as we've seen with the Stalk ELISAs, we only see a good response after the first shot in the inactivated group, after the 8-1 with ASO3 was given. And you see that there's reactivity across the board, basically from H1 to these bad HAs, H17 and H18, even some reactivity in group 2. For LAIV, we didn't see much movement. And so this is really fascinating because we get a huge breadth of this antibody response. And then when we give the booster dose, then also the groups that had first LAIV and then the inactivated vaccine induce an antibody response. And again, it's equally broad. So it was really nice to see that. Just to conclude here on this first part, for this study, CVIA 057, the adjuvanted chimeric HA inactivated vaccine induces high Stalk reactive antibody titers even after one vaccination. We think that might even be good enough for pandemic preparedness. The induced antibody response is very broad and durable. I couldn't show you the data here, but the induced antibody responses are multifunctional. We see Sinai, ADCC, ADCP, neutralization, even trimer interface antibodies. And if you do passive transfer into mice, we can protect the mice with the post-vaccination sera from these people who got vaccinated. We see that the adjuvant was important. The ASO3 is an excellent adjuvant, and the vaccine was safe, comparable to licensed QIV, LAIV, or H5N1 adjuvanted with ASO3. Just the overall conclusions about the chimeric HAs. Stalk reactive antibodies can be induced in humans by rationally designed vaccines. That's the first. A truly universal influenza virus vaccine will need a group 1, group 2, and influenza B component. And currently we are developing these group 2 and flu B components. And we think that in the near future, we will be able to test all of these three components side by side in clinical trials. But now let me jump to the second glycoprotein of the influenza virus. As I mentioned, there is a second surface glycoprotein, and that's the neuraminidase. You can see here in these electrotomograms how hemagglutinin and neuraminidase look like. HA has more like this peanut shape. Neuraminidase, the second glycoprotein, has more this mushroom shape. And typically we have less neuraminidase on the virion as compared to hemagglutinin. The ratios are often 4:1, 5:1 even. But the neuraminidase is important because it has an enzymatic activity that allows the virus to travel through mucosal surfaces and to bud from infected cells. So the Stalk domain of the neuraminidase has not been solved in terms of crystal structure, but the head domain has. The protein is a tetramer. You can see it here from the side and here top down. And so you have four protomers that come together, and each of these protomers has an active site that has sialidase activity. Importantly, only if you have a tetramer, you really get a fully functional enzyme. Now, why are we interested in the neuraminidase? How can immunity to NA contribute to protection and breadth of protection? As I said, NA activity is needed for transport of virus through mucosal fluids. There's a lot of decoy receptors that have sialic acid on our mucosal surfaces, and so the virus can get stuck there, and NA releases it, so it can go on to infect cells. And of course, it's also needed for virus to bud from infected cells. And these activities can be inhibited by antibodies. We have also seen that these NA antibodies have one component of protection that might work via effector functions. And this has been published widely, is that anti-neuraminidase titers in humans correlate independently with protection. And of course, NA is a validated drug target. For example, oseltamivir is targeting it. Now, NA antigenic drift is discordant with HA antigenic drift. They don't drift usually at the same time. And so that provides a safety net. If you would have a vaccine that gives you a lot of NA immunity, and HA immunity, if the HA drifts, the NA immunity might still be protective. Also, NA drifts slower than HA, which is also an advantage. And we have seen from monoclonal antibodies from humans that there is the potential of cross-protection to emerging viruses that have N1 or N2 neuraminases like H5N1 or H9N2. I won't go into the details here, but compared to the Stalk antibodies, neuraminidase antibodies are usually cross-reactive within a subtype, very rarely across subtypes. But within a subtype, you can get very nice coverage. Now, the current situation is that we have vaccines that do not induce strong antibody titers against the neuraminidase. This is shown here. We get a strong response against the head domain of the HA, not much against the Stalk, and very little against the neuraminidase. This might have different reasons, including small amounts of NA in the vaccine, unstable neuraminidase that falls apart, but there's also a component where you have immunodominance of the HA over the NA. This has been shown even back in the 80s by Ed Kilborn. And now the idea would be to solve this problem by either adding a lot of recombinant neuraminidase that's stable to the current vaccines, or use the neuraminidase on its own as vaccine. Now, these attempts have been made in the past. There's two ways to actually make neuraminidase, recombinant neuraminidase. One is by making it the way Flublok is made, just instead of hemagglutinin expressing neuraminidase in the bacular system in insect cells, and then membrane extract that neuraminidase. Protein Sciences has done that in the past. The disadvantage here was that the yield was relatively low and the stability was unclear. The other way to make neuraminidase is to just leave out the Stalk domain and the transmembrane domain, add a tetramerization domain, and usually a human VASP exogenous tetramerization domain is used for that. Express just the head domain where the protective epitopes sit, and that expresses very well and can be secreted into the cell culture supernatant. Now, the problem here is this VASP domain as tetramerization domain works very well, but it's a human part of a human protein, and so you don't want to vaccinate humans with that. Now, we wanted to work on that problem, but the first question that we asked was, do we even need tetrameric NA? And so we did a few experiments where we expressed monomeric or tetrameric NA. I can see here on the left side, for the monomeric, we even get a little bit of dimerization here. When we look at the NA activity, only the tetramer is active. Both are immunogenic. If you vaccinate mice, you get an immune response against both forms, but only the tetrameric form actually induces NA inhibiting antibodies, so kind of neutralizing antibodies. And then when you challenge these mice, only the tetramer induces protective immunity. The dimer is actually somewhere in between, but there's really a strong difference between monomer and tetramer. So the idea was, yeah, we need a tetramer. But we cannot use that human tetramerization domain, so we looked for other tetramerization domains that we could use. And we found viral tetramerization domains that could be used from measles and Senda virus phosphoproteins. And we also tested one from an Arabidopsis thaliana transcription factor. And so we made these constructs with N1. The positive control here is the VASP N1. And what you can see here is they look quite fine on the reducing STS page and the non-reducing STS page, but here we already see degradation for the Sinai phosphoprotein tetramerization domain. And then when we do cross-linking, so this gives you trimers, we see that the N1 with the measles phosphoprotein actually looks very, very nice. It's hard to see here, but we don't get really tetramers with that Arabidopsis thaliana tetramerization domain. And the Sinai construct is a little bit higher up here, and we might also get degradation. Nevertheless, we went ahead with the measles and the Sinai constructs and compared them more thoroughly to the WASP construct, and this is shown here. We have the enzymatic activity here, and you can see that it's relatively similar. If we vaccinate mice and challenge them, we get very good protection with all three constructs. And when we probe with monoclonal antibodies, partially broadly reactive NA antibodies, we see that the antibodies bind to all three constructs relatively well. But because we had this degradation with the Sinai protein or Sinai construct, we went ahead and further characterized the measles phosphoprotein tetramerization construct. And in collaboration with Ian Wilson, Ian's lab was able to actually solve the structure of that neuraminidase construct. It's shown here on the left. So this N1 construct that we use is based on Michigan 15, and it basically looks exactly like a similar neuraminidase from California 09, which is also an N1 neuraminidase. So we managed to make relatively authentic neuraminidase. I don't have to go into more of these details. There's actually a paper in submission. We'll have a lot more results on the recombinant neuraminidase out soon. But the overall conclusions here are that these recombinant A constructs with a tetramerization domain suitable for use in humans have been developed. In our case, that's the measles phosphoprotein tetramerization domain. We could show in the past that these recombinant NA constructs are highly protective in the mouse model and in guinea pigs within an NA subtype, and in guinea pigs, they also protect from transmission from one animal to another. We also see, and I haven't shown you the data, but the HA seems to interfere with the immune response to the NA. So if you mix QIV with the regular flu vaccine with a recombinant NA and give that to a mouse, the NA response is suppressed even there, even if you give a good amount of correctly folded neuraminidase. But we have found technical solutions to overcome this. You will see these publications hopefully in the next few months. And we started with N1, but we have also now worked on N2 and influenza B and A constructs that have been generated and which also look very promising. And with that, I would just like to acknowledge all the people working on these two projects. Of course, it's a lot of people. We have been working on this on broadly protective and universal influenza virus vaccines for a very long time. As a long list of people from my laboratory, people in gray left the laboratory over time. I have the feeling that half of my lab is now working at Moderna. And these projects are really a strong collaboration with Peter Blazes' laboratory and Adolfo Garcia Sastre's laboratory. Of course, we have many outside collaborators, including the clinical sites who evaluate these vaccines. And I would also like to thank our funding sources for CVIA 057. This was the Bill and Melinda Gates Foundation. The work was done together with PATH. And now for new projects that we started, the recombinant NA and the group 2 chimeric HAs, these projects are funded by CIVIC. With that, I'd like to thank you for your attention, and I'll be there later to answer questions. Thank you.